Hello everyone. So now I will start with the introduction part. Basically, this is the details of the article that we have chosen with the title of Molecular Structure of Nucleic Acid, which cover a lot about genetics code in our body, and the author is J.D. Watson and F.H.C. Craig. So what is DNA? DNA is a combination of two single strands forming a double helix structure as represented in Watson and Crick's model. DNA is self-replicating material that is in every living organism. In simplest terms, it is a carrier of all genetic information. It contains the instruction needed for organisms to develop, grow, survive, and reproduce. It is one long molecule that contains our genetic code. Therefore, for the biochemistry topic that has been covered is nucleotide and nucleic acid in chapter 2.
Before we start our discussion about the structure proposed by Watson and Crick, in this article, both refute the models of DNA proposed before them, notably the model proposed by Pauling and Corey, and also another similar model but a more detailed one by Fraser. There have been many models of DNA proposed after it was first discovered to have double helix in 1953. The double helix of DNA was based on the X-ray diffraction images of DNA, as well as base pairing chemical and biochemical information. The most famous of these early models was by Linus Pauling and Robert Corey in 1953, in which they proposed a triple helix with the phosphate backbone on the inside and the nucleotide bases pointing outwards. So, how was it proven wrong? Upon studying the model proposed by Pauling and Corey, Watson and Crick soon identified several problems with the model. First, the material which gives the X-ray diagram is the salt and not the free acid. The negatively charged phosphates near the axis repel each other, leaving the question of how the three-chain structure stays together without the acidic hydrogen atoms. Apart from that, the model proposed by Pauling and Corey has a big flaw. Some of the van der Waals distances appear to be too small.
I will talk about DNA structure that are mentioned in the article. First, DNA structure has two helical strands, each strands coiled around the same axis, which are called fiber axis or central axis. Two DNA strands are called polynucleotides, as they made of simpler monomer, which are called nucleotides. Nucleotides consist of phosphate, deoxyribose, and nitrogen-containing bases. So, DNA is composed of deoxyribonucleotides. They are linked by 3,5-phosphodiester bond. The strands are anti-parallel, which means one strand is in 5 to 3 direction and the other strand is in 3 to 5 direction. The bases are inside of the helix and the phosphate and sugar, as in deoxyribose, are outside of the helix. Phosphate and sugar is the backbone of the DNA structure. The diameter of the double helix is 20 angstrom or 2 nanometer, and the double helical structure repeats at the interval of 34 angstrom or 3.4 nanometer, which corresponds to 10 base pairs. The two strands consist of purine and pyrimidine bases that are held together by hydrogen bonds. The planes of the bases are perpendicular to the fiber axis.
From the research, they believe that the bases only exist in the structure in the most probable tautomeric form, which is with the configuration of keto rather than enone, and only specific bases can bond together. These pairs are adenine with thymine and guanine with cytosine. In other words, whenever one strand has an adenine, the complementary strand will have a thymine, and its cytosine is matched with the guanine on the opposite strand. The sequence of bases on a single chain does not appear to be restricted in any way. However, if only specific pairs of bases can be formed, it follows that if the sequence of bases on one chain is given, then the sequence on the other chain is automatically determined. They have found it experimentally that for DNA, the ratio of the amount of adenine to thymine and the ratio of guanine to cytosine are always very close to unity. DNA structure are impossible to build with the ribose sugar instead of deoxyribose, as an extra oxygen of ribose would make two close van der Waals contact.
DNA is a macromolecule consisting of two strands that twist around a common axis in a shape called a double helix. The double helix looks like a twisted ladder. The rungs of the ladder are composed of pairs of nitrogenous bases, which is base pairs, and the sides of the ladder are made up of alternating sugar molecules and phosphate groups. Covalent bonds join the sugar of one nucleotide to the phosphate group of the next nucleotide, forming the DNA strand's sugar-phosphate backbone. There are four nitrogenous bases that occur in DNA molecules, which is cytosine, guanine, adenine, and thymine. Cytosine and thymine are pyrimidines containing one ring. Adenine and guanine are purines containing two rings. So, to conclude, DNA is essential to life as we know it, replicating itself and passing its genetic information from parent to offspring.